Hi everyone, this is Ramalinga Prasad Kuppa. Welcome to my channel Pharma World. Today's topic is typical deviations in pharmaceutical manufacturing. In approved procedures for pharmaceutical product manufacturing, are deviations permitted? The answer may be no. So, how do you understand the deviation handling procedure? The answer is that if any deviations occur, it should be investigated for the causative factors. Based on the investigation report, CAPA should be designed. Let us see two examples in this video. One is production related and the other QC testing related. Deviation or incident. Deviation is a departure from the approved procedure. To understand in a simple way. So, if there is an accidental departure or unintentional departure from the approved procedure, it is necessary to record the deviation and investigate for the root cause. Deviations are classified as critical, major, and minor depending upon the quality impact on the process or the product. If the impact on the process or product is significant, it may be classified as critical or major. Impact means the adverse effects that are responsible for product failure. Minor deviations are minor in nature and do not significantly impact the product quality. Section 2.16 of ICH Q7 and 21 CFR Part 2 11.100b prescribes that all the deviations must be recorded, investigated, and conclusions drawn. These guideline sections are important to understand the requirements. Section 2.16 of ICH Q7 prescribes for the APIs and 21 CFR Part 2 11.100b prescribes for dosage forms. The concept is almost same in both the cases. Incident is an accident and that particular activity should be abandoned for further processing. Do not get confused with the other term incident. Incident is only an accident and further process is abandoned when the incident happens. Few examples are accidental spillage of input materials during handling of production, weighing errors while solution preparations in QC testing, accidental breakages of glassware during testing activity, etc. This is because there will not be any approved procedure available in GMP for such accidents. Obviously, if such incident happens, immediately it will be corrected and proceeded further. There is no impact on the end result of the activity. Typical deviations. Let us see one example in production area. The yield of the wet cake was marginally lower than the lower specification. The yield of a product wet cake was 98 kilos against a specification of 100 kilos to 110 kilos after isolating in a centrifuge. In such cases, the investigation should focus on the vital aspects. There is a clear clue on the physical loss of the material. So, the investigation should focus on the final stages of the isolation before going into phase 2 detailed investigation. Let us see. The deviation investigation should be as follows. This is a clear case of physical material loss. there is a loss of 2 kilos of the wet cake. The input clues for this deviation could be 1. The wetness of the cake, 2. The operation of the centrifuge, 3. The centrifuge bag integrity, 4. The input material charge, 5. The reaction completion before isolation stage, and 6. The cooling temperature for precipitation or crystallization of the product. Point number 1 is to check whether the product wetness was within trend or does it look more wet than usual. Point number 2 is checking the centrifuge operation. Was it performing as expected? Point number 3 was checking the integrity of the centrifuge bag. Was it okay? Point number 4 is to check if there is any possibility of lesser amounts of input materials charged when compared with the actual requirements. Point number 5 is to check the reaction completion test to confirm that all the product is isolated without leaving in the mother liquor. Point number 6 is also important. Was the cooling temperature as per the requirement? Now, the investigation should start with the following aspects. Is there any specification for the wetness of the cake? If there is a in-process test for this wetness, the investigation should go for the test. If it is water in the final product isolation, KF test is the best test that gives you the wetness content. If it is any organic solvent, LOD test may be done. 2. Is the wetness in this batch alone out of trend or abnormal? This is another point. Was there any abnormality in the appearance of the wet cake? 3. Is the centrifuge operating correctly as per the required revolutions per minute (RPM)? Was the centrifuge running with the required RPM? 4. Is the variable frequency drive working as per the design? The VFD controls the speed of the centrifuge. This also must be checked for its performance. Is the centrifuge bag intact without any damages? This is very common cause. While unloading the cake from the centrifuge, if care is not taken, there is a possibility of damage to the centrifuge bag and some amount of the product may pass into the filtrate. At this stage, the filtrate may be checked physically if any additional haziness was there. 6. Is there any evidence of spillage around the centrifuge while unloading? This also is possible when the operation is rushed to meet the targets. Sometimes it may spill out in the centrifuge area. Are all input materials charged as per the requirement for the batch? This may be checked in the BMR in the Bill of Materials section. 8. Is the reaction completion stage passed by IPQC? This may be checked in the IPQC release slip. 9. Is the temperature of the reaction mass maintained as per the requirement throughout the precipitation or crystallization? This may be checked in the continuous temperature record section of the BMR during the isolation stage if it is done manually and through a digital record if available. Is the product left still dissolved in the mother liquor? Firstly, the physical appearance of the mother liquor would indicate if there is any additional haziness or turbidity to establish the presence of uncrystallized product in the mother liquor. All these aspects should be considered in a systematic, scientific way in the investigation. All these observations should be well described in the investigation report. Don't go for yes or no type answers. The investigation can easily and effectively be done by using the fault tree analysis FTA as prescribed in ICH-Q9 Quality Risk Management Guidelines. ICH-Q9 is a wonderful guide for risk management. The FTA tool and its uses are well described in Annexure 1.4 of the guideline. A fishbone diagram of men, materials, machines, methods, measurement also helps to find out the root cause of the failure. This is another way to handle the deviations for any failure. A detailed risk assessment would help the design of effective CAPA for the failure. The detailed investigation using the above tools would give a comprehensive risk assessment report to design the CAPA. A low risk rate could indicate to improve upon practices. The moderate risk rating could indicate revision of some procedures to avoid recurrence of such failures. The high risk rating could indicate revising the existing system with another effective system, including training of staff. The CAPA should be based on these reported classes. Typical deviations, example 2. This is a deviation related to QC testing. The product failed to meet the specifications. Related substances test failed to meet specifications. The impurities were at 0.15% against the limit of not more than 0.10%. The failure may be due to two reasons: testing error and the product failure. As recommended by the guideline, it is necessary to establish that there is no QC testing error before going for a detailed production related investigation and further reanalysis and or resampling. Initially, it is necessary to negate the testing error. If there is testing error, it could be investigated and closed out with necessary CAPA. So, a detailed checklist should be used for investigation. The checklist should be used only as a guide and not as an all-inclusive checklist. The checklist and its peripherals must be focused. If there are no issues found in testing, then production area should be focused. If there are no issues in QC testing, the investigation should start with review of the BMR and the peripherals. The peripherals include calibration of instruments, preventive maintenance records of the process equipment used, approval status of inputs, training of staff on the operations, etc. The investigation should focus on the instrument. Mostly it may be HPLC that was used for testing. So coming back to QC investigation, in most of the cases where the failure was on the related substances test, the testing equipment will be HPLC. So the focus should be on the HPLC testing procedure. Following should be checked and recorded with detailed description. Are the calculations correct? Firstly, the calculations check. It may be necessary to recalculate if the report was okay. Are the glassware clean and correctly used? This is important. Certain analysis prescribes to use low actinic glassware. To make it simple, low actinic means amber colored glassware. Cleaning procedure is a very important procedure. Cleaning small volume glassware is really a challenge. You should never leave the cleaning of such glassware totally on the laboratory boys who clean the glassware routinely. Are the reference standards valid within the expiry period? This can be checked with the help of the label on the standards and reagents. Is the instrument in good calibrated working condition? The calibration status of the equipment should be checked. Are the test preparations and standard preparations made as per the approved STP? Pharmacopoeial test prescribes, as an example, prepare 1 mg per ml solution of standard and dilute to give a 0.1 mg per ml solution, etc. This is hard to understand. The STP should be clear and in an imperative way so that the analyst won't make any mistakes. The same can be written in the STP as dissolve 10 mg of the standard in a 10 ml volumetric flask with a specified solvent. Pipette out 1 ml of this solution, transfer into another 10 ml volumetric flask and dilute with the same solvent. This is easier to understand. The analyst will not make any mistakes. Are the system suitability parameters passed? The system suitability parameters decide if the instrument is suitable for analysis at that point of time. So, for each test, it is necessary to have system suitability parameters well established and within specifications. Is the integration done properly, auto or manual? Most of the integration is done automatically, but it is important to check whether the integration of tailing peaks or fronting peaks are not shaved or extended to get a different value. There should be a procedure for manual integration also in case necessary. All auto integrated peaks are not correctly integrated always. Are there any unresolved closely eluting peaks possible? The overlapped unresolved peaks will give more area leading to failures. This should be checked during the investigation. Are there any ghost peaks in the chromatogram that got integrated? Ghost peaks are another problem. It is necessary to check if any such peaks appear on the chromatogram. Is the analyst trained and evaluated on the test method? This is most important. The analyst should be trained and confirm that the analyst is capable of carrying out the analysis without any confusion. Answer to any of the questions above is positive. It is necessary to go into the root cause, and when once the deviation is confirmed, the test may be repeated and deviation closed with a kappa. After this, the kappa is designed. If there is any issue in the above investigation, the kappa should be based on the resolution of the problems noted during the investigation. Answer to all the above are negative. Then it is necessary to go into the review of the batch production activity starting from review of BMR. If all the answers were no, it can be concluded that there were no testing related issues and the phase 2 investigation into review of production activities, and necessary retesting and resampling may be done. It is important that the investigation should be descriptive and not to use yes no type tick marking method. As explained above, never use this format for investigations. The details should be descriptive enough so that the entire details are captured comprehensively. The investigation should continue into the other peripheral issues also to establish the root cause. The tools for risk assessment. The proposed CAPA can be effectively designed by FMEA or FMECA. If these tools are understood well, the assessment results will be very effective. Let us see. The most important tools that are prominently used are FMEA and FMECA. Conceptually, both are same and provide same information. FMEA classifies the risk qualitatively as low, moderate, and high. FMEA tool classifies the risk qualitatively as low, moderate, or high. Rating for severity, probability, and detectability are assigned qualitatively as low, moderate, and high. Then the overall risk is established. FMECA classifies the risk quantitatively by specific score for each potential risk, and finally the SPD score. FMECA tool assigns a score on 1 to 5 for harm, for the identified risk, for severity, that is S, probability, that is P, and detectability, that is D. The overall risk score is multiplication of severity score, probability score, and detectability score, that is SPD. One point to note here is for severity and probability, the higher the rating, higher the risk. For detectability, higher rating of detectability will have lower score and low rate of detectability will have higher score. Hazard analysis and critical control points HACCP, hazard operability analysis HAZOP, and preliminary hazard analysis PHA may also be used. These tools are used to lesser extent in the pharmaceutical manufacturing. While investigating deviations, it is important to revolve around the vital causes of failure with more focus than other trivial points. This is the core requirement. The investigation should revolve around the risk clues and not on other external reasonings. For example, if the yield is less and the suspect is centrifuge, don't look for issues with reactor. Focus initially on the centrifuge. It is certain to get answers there itself. Training for every failure is not a solution as kappa. The system has to be looked into for improvement. Many kappa reports do this mistake. Training is taken as a general item to include in kappa. It is not always the training aspect that causes the failure. There could be other system related issues for improvement and to avoid recurrence of such issues. I hope that the examples given for handling deviations reflect the routine issues in pharmaceutical manufacturing. The focus is that the investigation should conclude only till the root cause is established. In cases where the root cause is not at all clear, hypothesis investigation helps to determine the cause for the failure. Thanks for watching. For more videos, please do subscribe, like and share. Also, please leave a message in the comments box for any further support. Thank you.